the mute. Yeah, thank you. So I'm still not totally fit. Thanks so much for you organizing this, for you coming. What I want to do today is I want to have a brief overview of some of the things we do with a focus on this topic of personalized health because of obvious reasons. And I will put a strong stance in for machine learning. What our group is known for is simply the combination of evolutionary information and machine learning. That is somehow our niche. This is our group. You have seen several in this group. Today fewer because on top of all my disease, we actually have a couple of parallel events. So there's the other encounter. So we have a meeting with the Luxembourg group, and then there's another meeting that I have. And it's very difficult for me to be at 3 places at the same time. Now, those were in the room yesterday. Let's begin with this one here. So evolutionary information, the crucial picture somehow is the one that comes actually from Chrysander and Reinhard Schneider, who also was in the room yesterday, where what you see here is number of residues aligned, percentage sequence identity. What it means is whenever 2 proteins naturally evolve, they essentially populate the blue area. That means, for instance, let me pick a point, you have 100 residues aligned, and more than 33 here are identical. That means if these 2 proteins evolve naturally, they essentially have the same structure. Okay? Now this is remarkable. It's remarkable because if you look the other way around, it means you can change 67 in 100 residues and you're not changing structure. This does not mean that you can just take a residue and change it. Not at all. In fact, if you took 5 residues out of 100, most likely you would change the structure. Most likely you would change the protein. The protein would, most of the cases in which you just pick 5 and change them, most of the cases the protein would just not do anything, would not fold. Why then is it that we see 67 in nature changed without changing anything, and I cannot do it in the lab? And ultimately the answer is the neutral mutation, the one that does not change the structure, is very unlikely. But that's the only thing that survives. Now this is why in the track record of the structures that we see, we have the track record of survival. That track record, it cannot change at every single position, every residue. I have to carefully choose the ones I can change against what. Maybe somewhere I can change a positively charged residue against another positively charged residue, nothing else. Maybe somewhere else I can change something big against something big. It maintains somehow the biophysical shape, the physical features. And now this information, what I can change where, that's what you have in alignments of protein sequences that naturally evolved. That's what I mean as revolutionary information. And in this information, you have the information about the structure. Okay? It's all in there. It's a tiny bit of a problem. We can't get it out. We can't see it. So we can make some conclusions from alignment. We can learn some things from alignment. But we cannot stare at an alignment and see the structure. That's impossible. For that, we need machines. And machine learning is the first way in which that worked. So shown here for the case of secondary structure prediction, and I will actually come back to that issue of secondary structure prediction, where essentially this is a simple neural network, the input, the output, and for secondary structure, what you want to predict is helices, strand, and everything else. So you have 3 output units. And the input unit, in this particular case here, that's 13 consecutive residues. So for one window of 13 consecutive residues, you try to predict whether the central residue in that window here is, for instance, in a beta strand. Okay? And the new thing now is, rather than putting in the sequence information, you put the alignment information in. And that's essentially the combination of machine learning and evolutionary information, and that brought a big boost in the terms of the prediction of secondary structure and for many other things that were done afterwards. I'm just listing some. So let me get into the next statement that I believe in. Machine learning is the understanding of the 21st century. And for me, there really is, and that is an important part of my message, there's some phase transition that happens now. So 3 years ago, the world was different than it is today. Something is happening just now, and that has to do with the accumulation of data. that has to do with big data. It has to do with all of these hype words that you know. But suddenly everything comes together. So I've been working for over 25 years in machine learning in biology. And the success that I showed you is very old. I did that over 20 years ago. But still, the crucial way in which data comes now just brings it to a new level. That is what allows us these hype things of deep learning. But ultimately, there's much, much more than hype in there. Something is changing today. We are no longer going to have a machine do chess by just computing all the numbers. It will use machine learning. It will use a probabilistic approach. And that ultimately was Go that taught us that. Winning in Go by a machine over the world champion. That was one point in time where you saw a difference. There are many others that happened over the very, very recent span of less than 18 months. I believe these last 18 months just show something that is happening underneath. But the real thing is the big data. It comes together really now. Understanding. So let's begin with this one here. It is very clear when you do an analysis of data. So you try to see whether protein interaction is related to amino acid composition, or is there some amino acids that relate to protein binding, or some amino acids that relate to a particular disease. And all of these are analysis. The literature is full of that, and we all begin with this. And a lot of that is very important, but that is not understanding. The only way I can prove that I really understand the problem is if I can predict. So if I can say, okay, now I see this residue and I predict it's going to be that disease. I see this residue and I predict it's this type of binding. And if in some sense most of the cases, or better than random, I do right, then I did learn something. So in that sense, prediction is the acid test, the real important thing. But what, yeah?
I just asked, what is understanding?
So I obviously bypassed that question because I don't know the answer to that question. So I talk about, we're sitting in a technical university and I'm entitled to talk in an engineering type of environment. For me, then understanding means I can predict or I can do something with my knowledge. For instance, I know that there's a reaction with a chemical that I want to avoid. And I believe it's exactly that site, it's that residue, and if I change that residue, I will not mess up the other function of that protein, but only that drug binding or that chemical binding. Then I would argue that all the steps that got me to the point of I change that residue is part of my belief of I understand the problem mechanistically. I understand the problem of how it binds. And that understanding leads me to propose a change. If the change works, then I verify that I had understanding. I mean it in this practical way. Understanding in the sense of art, I can do something with it. Okay? Art, simply the Latin word I can do. It's the same thing here. But to which extent is machine learning helping here? And machine learning often is perceived as black magic, as the thing that is closed and you can't see anything, and that clearly does not give any understanding. But in my view, this is a total nonsense. This is a complete misunderstanding. And that misunderstanding comes from the simple reality: machine learning is complex, but it is as complex as the problem. So that implies that sometimes when you solve very, very complex problems, you don't understand the solution. That does not mean that you didn't understand. It means that the problem is so complex that you need a complex explanation. And the complex explanation, our simple mind or our mind tuned into finding fast shortcuts to understanding that are most often right in our life, doesn't work for complex issues. There is no shortcut for complex issues. That is the reality. And the machine learning matches that reality, matches that complexity. You could get the rules out of the black box. The problem is you don't understand them. They're meaningless. The moment you translate these rules to something that you understand, they are no longer reality. For my secondary structure prediction method, I did that, for instance, and I sort of recovered what people called understanding of how particular residues are related to forming helices or to breaking helices. But from these rules alone, you couldn't predict secondary structure at that level of accuracy. So yes, you can recover rules that you know, but these rules, in fact, are not the understanding. These rules fall short of doing technically the problem, right? They can't do it. That is something that we have to remember. It's not a black box. It's something that has inside a very complex reality, which is not, we cannot get the complexity out of it. Now, this does not mean that any complex, the more complex, the more stupid, the more likely it's true. This is not what I'm trying to say. So you can do a lot of damage with machine learning. And most likely, most applications of machine learning are mostly wrong and are very misleading. Still, this is true. Deep down, if you do it right, this is true. Now, in order to make this true, in order to make machine learning extract the truth, what you need to do is get a right cross-validation and the data set right. And I'm going to show you some of the typical things people expect to do this and some ways in which this goes wrong. It begins by simply, so the full circle in red is meant to be the training, the testing, in green, the training. So you have the full thing here, the whole pie is all the data that you have experimentally. You take, say, 3/4 and train with it, and you hide 1/4 under the table, and you try to extract the rules and see for the one that is different, how well did it work. And then you redo that 4 times, such that ultimately every single thing in your data set has been used for testing just exactly once. Not more, not less. So this, in fact, is something that right here is usually violated. How many times you split is not really important. That's a longer discussion. But that's not enough. Because people typically do that, and then they say, well, here's the result of machine learning method 1, and here's the result of machine learning method 2, and they use these test sets for that. And they say, because machine learning method 2 is better, that's the solution. But that implies that you use some knowledge, namely how good it is, to decide on a parameter, namely machine learning method 1 versus 2. That's wrong. You cannot do that. That means you need a third set. So I call it cross-training. There are different words out there. So the literature is full of mixing these words. Almost everybody uses training for the same thing, but all the other words, and then there's validation sets. But the important concept is very simple. You have another set that you use to optimize any other free parameter. For instance, are you going to use an SVM or are you going to use a decision tree? That decision has to be done on this yellowish or ochre set here, the cross-training set. I call it cross-training because it's, again, something that you use for training. So that's why I like this word, because it's intuitively very clear. And this here, you never use before you really finish your job. Okay, that, however, is also not enough. Biological data is always biased, and there are many good and bad reasons for that. So that means you have to first cluster. And when you go into training, testing, and cross-training, then you have to make sure that the sets are independent, so different. How you establish how far away they have to be, what is your establishment for percentage sequence and the e-values, or whatever that is, that again is part of the analysis that you have to do before the story starts. That is why the dataset creation is so important in this, and that's why it's so difficult once you have spent 3 years on creating the dataset, and somebody comes, I want to test my new machine learning device, and give me the data. You give away 3 years of work. So the major work is actually in this part. Anyway, so we cluster this, and that is still not enough. So we have done as clean as you can. But we're all very, very good at fooling ourselves. So in order to avoid that, the best thing we can do, I call it here pre-release set. So usually when you develop machine learning devices, it takes a long time to do this. If you do a PhD thesis, several years. So you start on the day 1 with a data set, and you use that, you prepare that, and then you get some results, training, cross-training, testing. But in those 3 years, or even 3 months, if it's only a master thesis or a bachelor thesis, new data has come up, and that's what you should use. Now, see anything you claim for your test set. Yes, you split your test set, but does it actually work for new data? Try to do that. And that typically costs some effort. So that's a sort of little bit younger version of Trisander of me, a healthier version of me. And this got us to a paper that a long time ago predicted a performance on secondary structure prediction that in fact one thing that was right is that the prediction of its accuracy is one of a few numbers that turned out not to be wrong. So we proved, because we spent a lot of time on it. And then we published another paper And I only show myself here because of something that will become clear in a minute. And we also published a number. And we did the same procedure. Everything the same cross-validation, the same pre-release, and it was total nonsense. It was a lie. And in fact, for this particular case here, it took a long time to publish the paper that said we were wrong. Nobody wanted us to publish that because many others had improved over that, and they were all also wrong. And they were very much in the way to speak up and say we have been wrong. So this was a 2.5-year submission paper. Nevertheless, it was still true. The reason why it was actually true is this was based on an estimate of low-level experiments that were just not accurate enough. It turned out that the prediction methods, in fact, were better than the low-resolution experiments, and that's why we overestimated prediction performance. In reality, when we got more X-ray structures of membrane proteins, it turned out that the prediction methods are way worse than we thought, and that still is a statement that is true. Nevertheless, I claim that prediction is the test for understanding. Now, again, in the practical sense, understanding, I can do something with it. I want to show you some of the challenges. Some already showed you. But before I do, let me turn this around and show you an example how you can learn from machine learning. Not only to solve a problem, but something that has to do with understanding that is beyond really, I can do something with it. And it goes back to, let me lead you quickly through the slides. Historically, how can I cut this? Okay, at some point there was a neural network that predicted secondary structure, shown here. And when I show over the training period of time here, that's learning, and that's the percentage performance, and the percentage performance for helix in red, for beta strand, and other. Now what you immediately see here is that beta strand learns slower. Okay? First the thing learns helices and other, and then slowly it learns the blue, and the blue always stays below all the others. That's what you immediately see. And in fact, so the maximum here, and you see there's sort of a green-red iteration here, the maximum here is already reached after one period of training, which is presenting every single example that you have in your experimental data once, and 10 times that, actually here nothing changes really. It only changes in the blue. Now, the method predicts beta strand very inaccurately at levels that are slightly better than random. And that was true for all the methods that existed at the time. And the argument was when you form a beta strand, you bring together parts of the sequence that are very far away in sequence space. The neural network sees 13 consecutive residues. But the beta strand forming hydrogen bond is not within those 13. While when you have a helix here, the hydrogen bond that determines the helix is within I and I+4. So that is within the 13, right? So the information you need to know, there's a hydrogen bonding partner, is in there, in the sequence for helices, but not for strand. That's why the field, all the experts of understanding protein structure had been arguing for decades. This is why beta strand prediction can hardly be improved, because the information is not in there. And we have somehow to bring the global information in there. And that, to some extent, is true. But another way of thinking about this, for me as somebody doing machine learning, it does not learn that fast enough. And maybe it does not learn that fast enough because I don't have enough examples. Actually, if I looked at now the whole pie is all correctly predicted residues, helix, strand, other, you see that the smallest number of the correctly predicted residues are beta strand. This is how many are occurring. Beta strand is only about 23% of all the data. And what you see is your accuracy pi and your occurrence pi look suspiciously similar. Now, if you mix that again with this observation that you learn beta strand only very, very, very slowly, you may suspect that the problem really is you don't have enough of it. And that you can circumvent by just giving, artificially enriching the class. So over-presenting beta strand. And that is shown here. When you over-present beta strand, now you give one example at each time point, then that's what you get. Exactly the same thing. Biophysically, the reality that beta strand formation is not local is totally convincing. But that is not the explanation. So here we learn from sort of a better understanding of the machine learning device to completely see that something that is intuitive and makes a lot of sense and explains reality is not right. Okay? Now, how we can use that for better prediction, that's a different story. It is usable, and just to show it around, this is an equal distribution, and if you present them equally, then that is a prediction accuracy. You predict them equally. The story is a little bit longer. So the second challenge really is the data set preparation. And here I want to show that on the example where it blows up the most, which is protein-protein interaction. For me, protein-protein interaction, so that's HIV, the HIV glycoprotein 120, GP120, here in green, bound to CD4 and the Fab antibody. And when I talk about protein interaction, I mean that two regions here touch each other, and those two things that touch each other come from two different proteins. Okay? So I would not call the interaction here between the different parts of CD4 a protein-protein interaction. That's the inside of a protein interaction. And, okay, so let me cut those facts. Now, I said in order to distinguish between training and testing set, I have to group. For protein interaction, this gets a little bit more complicated because now what I have to group is actually the pair A-B. So protein A and B interact. So now, in fact, I have to sort of do my clustering not by A's, but by the binding reaction. And that makes it slightly more complicated. In fact, what this one shows, this is recall versus precision here, and two different methods that predict, method 1, method 2, that predict protein interactions from sequence alone. And I want to say the green here is that the protein that I predict has been used for training. And the protein has not been used for training in green, has been used for training in red, and the same thing here. So the red is a little bit less higher here with respect to the green than here, but still, for both it's true that I want to predict A and B bind. I do not put the interaction AB into my training set, but I put one of the two, say A and C. So we don't have a pen, do we? Or marker? No? So again, I have A, B in my training set and AC in my test set. AC is a different interaction than AB. So you may argue that it's clean behavior because from the fact that A and B interact, I do not know whether A will interact with C. In that sense, keeping it in the training set is okay behavior. But it turns out this is a better approximation of truth. So I cannot keep it in the training set. Okay? So I have to take these two complexes apart and make sure that none of them has been used in training, even in context of different protein interactions. That is counterintuitive. Now here comes the next statement. So far I say I have to exclude A if it has been in a different interaction. But so far I've only talked about the positives. I've only talked about the proteins that interact. What about the ones that don't interact? Do I have to do sort of the same data set split and the same distance split and overlap split and bias analysis for negatives? And the answer for that one is true. We have to also consider the negatives. Otherwise we get the wrong results. That again was very, very, very counterintuitive for us. There was another statement here. If in the device that I'm going to briefly flash to you, we need all the data of protein interactions we can get. So should we take, if one protein I know is involved in many other interactions, and all of those have a lot of good data, should I take one protein more often because it's involved in more interactions? And the answer again was no. Sample one protein only one at a time. Again, very counterintuitive because we know that we don't have enough data. But oversampling this one just gets you completely into overtraining. And oversampling the data. Thank you from me. I just wanted to make this point again clear. It may be simple. So A, B is in training, and AC is in testing, and C is completely not like A. And that is not possible. And it's not possible in the positives, and it's not possible in the negatives, and you should not oversample A in any way. That is what this slide says. So there's a lot of stuff underneath. Ultimately, what it means is that the cross-validation analysis is getting extremely complicated for protein interactions. In some sense, I call it the problem is squared. Now, then there is a method, and it comes from Tobias Hamp, and all of this analysis comes from Tobias Hamp. And actually, one important aspect of that is that underneath, we have a method from Tanya and Tobias that predicts subcellular localization for eukaryotic protein at a relatively high level of accuracy using profile kernel SVMs. Anyway, so we can predict protein-protein interactions from sequence. Not very accurate, but it's the most accurate thing that you can have for this thing. But, so the first but is, it was a long, long way to get there. This is as long as it actually took us to develop that method that we just published last year, that Tobias published last year. But here's let's call it but 2. And the but 2 is method number 41. So method number 41, essentially it takes the positives from the PDB. So I know that protein A and B interact. I use high resolution structures for that because I'm very sure about that. How do I sample my negatives? Well, my negatives, I sample anything that nobody has ever observed in a protein interaction. Mass spec, whatever the yeast 2 hybrid, I don't care about the resolution. Anybody has ever said that these two proteins interact, I don't call them negatives. But nobody has ever observed anything, I call them negatives. Is everybody happy with that? Okay. This particular case gave us many, and we got method 41, and it was extremely good. And there's a tiny bit of a problem in method 41. And the tiny bit of a problem is it can predict whether a protein is from the PDB or from Swissprot. We did not expect that this was possible. I'm sure I can predict PDB versus the UniProt. I'm sure I can predict Swissprot versus UniProt. But that we can actually distinguish between PDB and Swissprot, I did not know. I'm very proud to know this. It's not quite true. This took, I mean, it's worth, you said 500,000 euro or dollar. This way more money got into this method 41 here. And you may very much wonder whether it's worth the money. And it's just one of those moments. I had not expected that. And had it not been for Tobias, I would have never found out. We clearly would have published this. Because checking something like that, whoever thinks about that? So we were extremely lucky that we got away unscared, yeah?
Were the PDB sequences pulled out of the PDB files themselves, or?
No.
From Swissprot?
Yes.
Yeah. Otherwise it would be obvious.
No, but I still, yeah, well, no, but you see for those of you who are not in the protein world, so UniProt is a database of all the proteins that have no annotation whatsoever. Many of those don't exist. Many of those, the protein sequence assignment is wrong. While Swissprot are proteins that have been experimentally worked upon. These typically are shorter. That's statement number 1. These typically are subsets of UniProt. Okay? And the same is true for PDB. They are also typically subsets. Okay, PDB is a little bit shorter even than Swissprot, but other than that, both are proteins that really exist, and I would have expected that somehow, I did not expect that they are so different, but they are.
It's a crazy idea, if it's worth looking into. Is it a matter of intrinsically disordered proteins, which obviously will never be PDB, and we know that it's possible to predict.
That's not true. So actually, that's not true. So most prediction method for disorder pick out a lot in PDB. Because they have been trained on the missing coordinates in PDB.
Oh, yeah. True.
Yeah?
True.
Can you clarify this for me? So obviously the PDB structures are indicated in Swissprot too. So what you need is you predict those proteins with the structure in PDB versus those proteins in Swissprot that have no structure in. Okay.
That's correct. So essentially it's a number thing, right? This is 100, this is 500 or something like that. And this, not all of these 100 at that point at which Tobias got his data set were completely in Swissprot. So the overlap is, there's some unique on that side too. Yeah. Okay, but anyway, so this was not, I did not want to say that we have a prediction method to distinguish PDB and Swissprot. This is not, we never published this, and this is not really important. What is important is it's very easy to go wrong. Okay, and 25 years of machine learning, we have never been that wrong. Now, let me give you some results in flashes. And this, I don't quite sure know how to do this. Edda is a major person behind it. Alistair Forrest and Jordan Romilovsky are the people here. So what they looked at is secreted proteins in this writing data here. And they find that receptors are cell type specific, and they often evolve, targets often evolve before the receptors. That's an amazing reality. The reason why I bring this up for you guys is essentially this is a little bit of sequence analysis of expression data plus annotations. So whatever annotation information you put in that is valid can help, and this is a high-level publication here, so you can contribute to exactly this kind of analysis. Okay, that's one of the reasons why I pull this up. Here's a different story, and that is the opposite. That is a story from Tanya and Jana that has just been accepted. So when antibacterial defense, they shoot out proteins to attack other bacteria. And now the question is, we know we have studied a lot of antibacterial defense. Now the question could be, we know some of the guns, so the way this defense is shot out, but we don't know the bullets. We know many bullets, and essentially those are the green ones here. And the question is, how many new bullets will be out there? So can we just rely on BLAST? And what briefly this says here is, if we are anywhere near right, or Tanya and Jana are anywhere near right, their predictions are blue here, then there's a whole universe out there that has never been seen before. So BLAST is absolutely not enough, and text mining in this particular case is absolutely not enough. The way text mining can help here is essentially machine learning in the sense that Juanmi presented yesterday. So get some text annotation, apply the machine learning, and see whether you sort of can step by step get higher. So they started with a very good experimental data set that they had from an experimental collaborator. Here's another story, and it comes from Debbie Marks and Chrysander. This is no longer true, so they're both at Harvard Medical now, and Thomas Hopf, who is also at Harvard Medical. This was the first paper in which protein structure had been predicted from sequence on a larger scale and has been ever since verified for many of those. So in their paper here in 2012 now, in Cell, they predicted 11 membrane protein, or structures for 11 membrane proteins, and at least 4 of those have by now been experimentally verified. None of them could have been done by any other method. And that never happened before. Membrane proteins are very difficult to do experimentally, and they're also very difficult to do in terms of simulation. And the reason why this was possible is, again, because they could look at very large data sets, the coevolutionary signal, and this is again something coevolutionary so you in some case of in some part of the protein in the alignment, say you have a big residue, and then suddenly you change to small. And somewhere else you have small, big. And maybe you see that here this change meant that there's a little bit of a hole, and the hole is filled by another change. So somewhere else the big one is the small one is changing to the big in that family, which means that actually the binding is the same. There's a small next to a big residue. It's here and here, it's the same. But then you suspect, if that is correlated, that they're actually sitting close in space. So seeing these kind of changes, you predict proximity, from proximity in terms of 3D structure. From this proximity, you can predict structures. That's essentially the idea. And that idea, again, had been around for over 20. So one person who is in the field of text mining has pushed very much here, Alfonso Valencia. And ultimately, it never really worked because there was not enough data. The breakthrough here, there was an idea underneath that was new or had never really been appreciated before. But another important point was the increase of data. So In the initial method, they needed 10,000 proteins in an alignment. Now they have refined that, but these are really large alignments that they needed, and that was the reason why they could only do it for 11 or 17. Okay, now that was already mentioned, the intrinsically disordered proteins. Another thing that is very, very important, for which we need actually more text mining, is this field of intrinsic disorder, because it's very difficult to get by experimental information for that. And disorder is very important in eukaryotes, so there's absolutely no annotation without looking at disorder. So here just one example. So disorder is in at least every third eukaryotic protein, while arguably only in every 10th prokaryote. So it's less important for prokaryotes. Still important, and what this work from Avner Schlesinger and Esmeralda Vicedo essentially says is that if I look at two proteins, at the disorder content of the entire organism, and I ask, are they more similar in terms of the disorder, or are they more similar in terms of the family? It turns out that you're more similar to the one who lives in your environment in terms of disorder content than you are to your relatives, which is surprising. But it tells you something about the adaptability. So it's like you sort of move into a different environment and you put on a little bit less or more disorder and you fit. And that's remarkable. It's not causal, but it's a surprising correlation that is observed here. Here's something that is causal, but I don't want to get into that. What I do want to briefly mention a little bit longer is, two slides longer, is work from Sean O'Donoghue. The slides come from Andrea, who was in the room yesterday too. And Nelson Petillayo is another person on this. You guys are in the wrong room, up there. And it is the work on the dark proteome. So what is the dark proteome? This the term Sean introduced. White is, I know experimentally, the structure. So this is one particular protein, and there are regions for which you know the structure. Then there are regions where you can possibly model a structure, because it's similar to some other structure that you know experimentally now. And so they are white, meaning completely experimentally known. They're sort of gray, means I can build a model or have a low resolution structure or something that is not. They're dark, absolutely no 3D knowledge, okay? And then when they look at dark proteins, and this is now, the entire protein is dark, and the percentages here are valid for SwissProt, so this is not, in that sense, it's a biased data set. All eukaryotes in SwissProt that are completely dark, so I have no knowledge about anything with respect to structure for that protein. So you see that fraction is disordered, that fraction is compositional biased, that is transmembrane, and these are sort of what structural biologists would call the common suspects. They are in the way of structure determination. Those are features, if you have them, it's difficult to get a high resolution structure. So this is the kind of thing that is surprising. How can you have something that really looks nice and still don't have a structure? That is, you may call the surprise. This is sort of the expected. Okay? And here's another thing about numbering. So one way of looking at it is you could argue, okay, roughly 1/3 of the story dark is explained by disorder. Right? Do you agree? Is that, if that's the fraction, so this is all the disordered proteins, all the dark proteins, and about 1/3 of those are disordered. Does that mean that disorder explains 1/3 of my cases, of my observed phenomenon? And here's why not. Now I look at the non-dark ones. And you see that, okay, the number is lower, but not by much. You have to remember what is background random, okay? Don't forget, don't only look at one side of the coin, always look at the rest of the universe, yes?
Are those completely disordered or only in certain regions?
Yeah, that's exactly the point that Jens made. So in PDB you wouldn't predict disorder, he said, and somehow you do. So this is entirely PDB here, and there is some disorder in there too. They are not entirely. That's Exactly. And again, it's only regions here. But the point is here, it's the same prediction method. So whatever it is, it doesn't differ that much. So the message is, yes, it is a significant fraction here, but it's not the one that explains it.
I mean, Esmeralda always took these proteins which have long regions, smaller regions.
No, it's not the case here.
These are complete, I mean, more or less completely.
No, sorry. This is the other way around. So you have two residues that are disordered and they count.
Okay.
So, right. Right.
Also at the terminal.
Yes, exactly. Also at this terminal. So you may argue whether it's right or not. And I don't want to argue this. The only important statement is that these two, this is entirely structurally known, this is entirely not known. What it shows is that by all I can say in terms of structural features, they don't differ that much. So that implies that in fact the reason why things are dark are not very well understood. And the idea that dark is transmembrane, dark is disorder, this is very misleading. Yes, it may explain some of that, but it's a tiny fraction. And most of why dark is dark remains dark. Unclear, so to speak. Okay, let me finally get into the personalized health story that went to a TEDx talk that was tough for me. We differ by 10,000, roughly 10,000 amino acids, 10,000 letters. And in the Bavarian coat of arms that in the Oktoberfest you may see again, essentially every pair in here differs by the red in the blue, right? Now, it may strike you as a lot, but given that I have 67 billion cells or something like that, and they all have the same essentially the same DNA, this is actually a surprising difference. And the question then becomes, do these differences affect us? The 10,000 by which we differ, does it matter? If we waited for the experiment, and that's another reason why I bring this up, this is another point where text mining is really needed to give better annotations and do some of that. Today, in order to answer the question whether they matter or not, we would have most likely to wait until 2015 or longer. Today we can do it in the computer, and the method that we use has originally been developed by Jana Bromberg and Max Heck. It's a neural network that predicts whether a particular change, amino acid change, leads to an effect or not. Effect could be that the binding gets stronger, or effect could be that the binding stopped. We cannot distinguish that. Neutral means essentially it's the same, okay? And what I'm going to show you is a couple of graphs that essentially sample cumulative values. So what I show, that's the score for the SNAP, the prediction method. 100 means strongly predicted to be effect. Minus 100 means strongly predicted to be not an effect. So if I had, and I'm going to essentially look at cumulative plots, meaning what fraction in my data set is predicted above 75. So if I had a data set with effect variance, and if my method would be absolutely right, then this would be here somewhere, right? It would essentially be a straight line. Everything would be predicted as an effect. If I started with a green set that doesn't have an effect, everything would be predicted to be neutral, if my method were right. Now, in reality, that's not as good as the method is. So this is for a data set of effect. This is of a data set of neutral. The distance to the end here is the mistake the method makes. And the degree to which it pulls these two curves apart is the power. So it's accuracy, right? It's a performance. Okay. Now, the first question that we can ask to that method is, if I start with residues that I know are monogenetically causing disease, so any carrier of that variant is going to have that disease. Okay? How, and they have never been used by the method. How is it going to be predicted? And the surprising reality is this is the OMIM variance here. So it's surprising because in machine learning, I never encountered a situation where I came with a new data set, and my performance that I'd never seen before, and my performance was better than I'd estimated. So that's the cross-validated result here, the red. And I do better. And the reason why I do better is because these data are so much cleaner. So when something causes a disease, it's absolutely a clear effect. And when I sometimes measure that something is an effect, there's more doubt about that. So we can predict that. And so this is work from Jonas that says for animal diseases, this overall number goes up a little bit even. So we can predict these single immunogenic diseases in animals even better. So now we can address the question, how do they matter? The 10,000, any pair in the room has 10,000 changes. How do they matter? What do you believe? Do they matter or not? So again, the data set that I'm using here is people like everybody here in the room. We are all healthy. Yes, I have still fever, but I'm healthy in the sense that I walked in here. And this is true for everybody who's been sequenced. So what do you expect for the entire data set? So will it be more toward neutral? That's what everybody expects. And somehow the difference that we see is in between effect and neutral. So this may be saying, yes, it is more toward neutral. And in fact, if we look at it in more detail, I will not give you the time for that, this is less than random. So it's a little bit more toward neutral than random. But not much, surprisingly not much. Now, if we looked at variance between us and Neanderthal, in this particular case it's not Neanderthal, it's the Denisovan, then that's the curve. So the Denisovan curve is a little bit sort of, it's not a clean curve because we don't have enough data. Okay, there's a sequence from Svante Pääbo in Leipzig and Janet Kelso for essentially one individual. And that's why this data is. Okay, let's just look at gorilla. There we have a lot of data. That actually for the first time is we have a data set that is below neutral, the cross-validation. So now I have to immediately say, I'm sort of changing the data set again. When I compare the data set between us, we use the 20,000 same proteins, right? When I compare us to chimp, or in this particular case gorilla here, we are not using the same protein. We are using the subset of all the proteins where I can make the clear statement that the gorilla protein is an ortholog of the human protein. Okay, it's a smaller subset than 20,000. And then I ask for all the variance between gorilla and me. What's the effect? But it's still surprising that this is more neutral than any pair in the room, right? And one thing is clear. The genetic differences that we have, the 10,000, they do matter. Obviously, they are not the killer mutations, but they clearly matter. Then we may argue, well, the ones that they matter, but they do not happen often. And that's wrong. And let me not get into that. I'm going to go through that. So how can that be? Ultimately, I believe what we see here is between people, we have a sort of living population. And in a living population, you accept the risk of doing damage because you could do well. And if one of us, under certain conditions, will survive with that variant, then this will be an advantage for the population. And that's my explanation for this event. Now, personalized health, then for me, is the attempt to some of those that in fact differ between us have a bad effect and some will have a good effect. So let's use personalized health to change the bad versus good effect in the ones that matter. That's my credo here, and I did not have enough time to really develop this, but I believe personalized health will help us to turn many of the variants between us matter, change something. Some of those will be bad changes, some will be good changes, and I believe personalized health will help us to focus more on the good ones. Conclusion. Our genetic differences differ. They contribute to our individuality, but also to our susceptibility of problems. Possibly bad for us, possibly good for our offspring. Personalized health, for me, somehow is to find the good in both worlds, to benefit from the diversity of us, and to somehow let only the good survive and keep the bad in check. Now, I believe really, I said that before, in machine learning we have come to the point of phase transition. The data accumulation is such that we now, the electronic usage of data, and again, that's an important part where you contribute to with text mining too. This is a phase jump, a leap, and I believe it's really, really good for the field. In terms of personalized health, and that's a recent paper from Predrag Rivoyats and Rihanna Bromberg, where they argue that we can use machine learning and clever uses of machine learning to in fact learn from the variety of the genomic data to go in between the two extremes we have today. So on the one hand, we have today what I just presented, the analysis where we can look at all the 10,000 variants. I can say something, but it doesn't really help my health, this analysis. It's an interesting statement. Then we can look at the ones like this one here, historically. Here we have one particular variant. You change one, the E to at position 7, so historically it's called position 6, to a V, and you cause a disease, sickle cell anemia, very bad disease. The same variant, however, helps in malaria resistance. So it has the same, this was in fact the first molecular change characterized by Max Perutz 60 years ago now, related directly to disease. And we understand a lot about this. For some of these residues that we know have an effect, we have this level of understanding. For the vast majority, we have this level of understanding. And we need machine learning to connect these two. Okay? It's going to be machine learning plus efforts in some sense like the one you do. We need a lot of annotation. And we need it today. Let me just put one step in there. You have a question? No. Keep the fun and enjoy teaching. So this is about a group of computer scientists wanting to learn JavaScript, data mining, machine learning. And the big idea was really put biodata in there. And you will give them a lot of fun. And that totally worked. Not at all, actually. Because ultimately, the whole room was occupied trying to understand the biodata. And that was very, very frustrating because it was perceived as too complicated. So then the big idea was, let's try something like this one here. So for those of you who can't see that, science versus everything else. So this says simple answers here but wrong, complex but right. Okay? So this is the bifurcation point, and everybody knows where most of the time we walk. They are as intelligent as most of us most of the time. And we thought about benefiting from this by doing something like that. So find something that is simple, not necessarily true, but something that the students really care about. We need something else, and the idea was use Game of Thrones, right? And a Game of Thrones simply predict who is going to survive the next show. And that was done in April, just before the new show came out. And they essentially predicted who...
Don't say, I haven't seen it.
I'm not going to... So...
Here's a clear funding opportunity.
So you have no idea. So this went skyrocket, and I show you some. So this is the most impressive one. 1.2 billion readers of this work in the way it has been announced in the press. And then all these TV people came to me. I had never seen Game of Thrones. I've never read it at that point. That was April. And all they ever wanted to know, who's going to die next? I wanted to talk about machine learning. I wanted to talk about all kinds of who. Meanwhile, I've seen it. But I'm not going to give it away. Anyway, the point I wanted to make here, this actually feels good. We have to put that in the background. So you're sitting at the Technische University of Munich. At the Technische University of Munich, it took them 15 months to build a totally new car. So they invited the press and said, we are going to have a new car. How's it going to look? We don't know anything. They had Amazon boxes, and they said, this is going to be the size. They knew nothing about it, and they said it's going to be new. And 15 months later, they presented that thing at an auto car exhibition that had 160 kilometer range, drove 120 kilometers per hour, and the whole thing on a 15 kilowatt engine. And it looked nice too. Okay, this was just a model. But this is what this university sort of can do, and I felt immensely proud when I saw this thing, that had not even 1/4 of that press coverage. So that's a really good thing they did, right? 1/4 of the coverage of this. The Game of Thrones, 4 times more. So it's an amazing, stellar thing. I'm saying that because I seriously mean we should do this kind of thing more often. We are sometimes too bogged down by the push of problems. And that was a lot of fun. And not only a lot of fun, a lot came out of it. I'm way over time. Thanks to many in the group. Edda is here. I thank her because of that most, simply because she's sitting there. Juanmi will come back in a moment. Many others, and here again, in particular you for organizing this, and several others here who have helped the group over the years. And thank you for your attention. Sorry for being so much over time. I still have a little bit tough time for questions. Then you have time for do we have a coffee break now?
Yeah, we have now discussions and we will do some working together. But, yeah.
Thank you for being in Munich. Enjoy your stay.